Hello, my name is Patrick Gallagher. Thank you for coming to my poster presentation. For the past 2 years, I've been working with the graduate student Lexi Hoopman in the Raymann lab. And today I'm going to be talking about how antibiotic exposure is lethal to developing male honeybees. To start off with, it is commonly known that honeybee overall health has been declining for over a decade, but their reproductive health has also been declining. Drones create all of their sperm during their development at the beginning of their life. The queens are the sole reproductive unit in the hive, and they will be laying all of the eggs. And finally, the worker honeybees, which are the female honeybees, do all of the hive tasks. It has also been recently found out that drone sperm storage is highly variable with no known cause. We also know that queens mate early in their life, so they are going on these mating flights and collecting highly variable sperm from these drones, which can lead to increased rates in queen replacements. Increased queen replacement rates can lead to an increase in hive mortality. Oxytetracycline is the most commonly used antibiotic in beekeeping, namely to treat bacterial brood diseases that need constant antibiotic exposure to keep the spore cells dormant. And finally, a typical field dose of oxytetracycline consists of 3 separate treatments, and we estimate one treatment to be around 200 micrograms per milliliter per bee. So this is the concentration that we will be using in this experiment to start off with. The objective of this experiment is to determine what concentration of oxytetracycline is lethal to drone larvae when they are reared in vitro. We hypothesize that the exposure of oxytetracycline during drone larva development will negatively impact their growth and their ability to reach pupation, which is the next developmental stage. To test this hypothesis, we first trapped the queen on a frame for 24 hours. This allowed us to know where she was laying the eggs and how old the eggs were. After releasing the queen, we allowed the hive to raise the eggs for 3 days. After this, we moved the frame into the lab and grafted them into one of 3 pre-prepared 48-well plates. Each one of these 48-well plates had one of our pre-prepared diets. Our control diet consisted of half water and half royal jelly. Royal jelly is the substance used to rear larvae in the hive. We then had our oxytetracycline diet, which consisted of the control diet and then a specific concentration of oxytetracycline that we started off with our estimated field dose. Finally, we needed an ethanol diet because oxytetracycline must be dissolved in 70% ethanol. Drone larva survivability was calculated each day as they were moved to a new 48-well plate and fresh diet. After 7 days, the larvae that survived were moved to a pre-pupa plate, and here the experiment ended and all final survivability data was collected. And here are some pictures that we took along our experimental process. The first one is showing the grafting tool that we used to move all of the larvae. The second one is showing day 0 larva. The third one is showing day 2 larva. The third one is showing day 5 larva. And finally, the last one is showing our pre-pupa plate. After our experiment ended, we were able to create a survival curve. We have the survival probability on the y-axis, and on the x-axis, we have time, which we're showing in days. Our control and our ethanol control diet treatment groups follow a very similar trend, indicating that the addition of ethanol had no significant effect on larval survivability. The 200 micrograms per milliliter, which is our estimated one treatment of the field dose, those larvae died within 4 days of exposure. 100 micrograms per milliliter treatment group died within 7 days of exposure. And finally, 50 micrograms per milliliter treatment group survived until pupation, but there may be sublethal effects that we could not see in this experiment. And here's just a quick rundown of the results that we just went through. And to highlight that the 50 micrograms per milliliter was the only group that survived until pupation, and that they are just a quarter of the estimated field dose that we calculated. And we also know that developing drones are highly sensitive to antibiotic exposure, which could lead to a variable quality in drones. And this varying quality in drones could be a contributing factor of colony collapse disorder. In the future, we plan to investigate those unknown sublethal effects of oxytetracycline. And we already know that antibiotics have adverse impacts on other animals, such as mice and rats. To investigate these sublethal effects, we plan to rear a new batch of drone larvae, starting with 50 micrograms per milliliter and raising them to adulthood. From here, we will look at their gut and reproductive microbial communities, their sperm viability, and conduct mass spectrometry to see what concentration of antibiotics reaches their internal organs. Finally, we do not recommend stopping the use of antibiotics as they are highly important in treating those bacterial brood diseases we talked about earlier, but we should be actively looking for an antibiotic alternative. Thank you so much for coming to my poster presentation. And if you have any questions, I'd be happy to answer them in the comments below.